So I am Lindsey Shartell. I'm the acting Forest Wildlife Research Group leader with our Wildlife Research Unit in the Division of Fish and Wildlife at the Minnesota DNR. Today I'm going to talk about measuring wildlife populations in their habitat with an emphasis on the wildlife population side, since I know we're going to hear a lot about habitat over the next couple days. So let's start off with a few definitions to set the stage. A population is a group of animals of the same species occupying a given area at a given time. Habitat is the resources and conditions that produce occupancy, including survival and reproduction by a given species. When we talk about measuring, that's simply ascertaining the size, amount, or degree of something. Whereas monitoring is repeated measuring of a parameter over time, the important part being that it's repeated over time. So why measure or monitor wildlife populations? To conserve wildlife, we need to know something about the population, its status and trends, when action is needed to protect the species and their habitats. Harvest management is determining the bag limits and harvest regulations, which requires knowledge of the population. Data on harvest levels and the resulting impacts on the population is necessary for ensuring a sustainable harvest. Public interest is another good reason. Measuring populations allows us to communicate with the public where they have the best opportunity to harvest or view different wildlife species. Legal issues. Determining the need for listing or delisting of threatened or endangered species often relies on population abundance targets, which requires population monitoring. Historical value, relating current conditions to historical population ranges and levels can help provide us some context for future direction. Species and habitat interactions. Understanding the response of wildlife populations to change in habitat and how changes in forest management might impact habitat and thus impact wildlife is particularly important. And then impacts of environmental factors. Understanding land use changes, climate change, and an almost endless list of other things that can impact wildlife is really important for being able to manage them. So like I mentioned, measuring wildlife populations can answer some of those basic information needs that we need to manage the species and their habitat. Simple things such as how many are there and where are they? Is the population increasing or decreasing? Are harvest levels sustainable? And is management action needed? It can also help us answer complex information needs. Are current practices or management effective? How do habitat changes affect the population? How do species interact to influence each other? And what outside factors impact the population? What are the mechanisms of a species' response to management? And of course, why? A good example here is the spring deer density in wolf range and the winter wolf abundance. You can see that there might be a connection between the two. So there are a number of different population metrics that can be collected to tell us something about a wildlife population. Depending on the species, your question, the logistics, you might collect one or many of these different metrics. Abundance is the number of individuals. For example, the estimated moose population in Northeast Minnesota. Density is the individuals per unit area. Occurrence is the presence or absence of a species. Range is the extent of species occurrence. For example, the wolf range and how it's expanded over time in Minnesota. Age structure is commonly the number of offspring per adult females, like the moose calf to cow ratio, though you can measure quite a few different things related to age structure in a population. Genetic variation is a particularly important metric for small isolated populations. and then vital rates such as births and deaths, survival and mortality. So it's typically unfeasible to obtain a total count of a wildlife population, except in very rare cases. So scientists use a population estimate to approximate the total population, understanding that there will always be some uncertainty around this estimate. When the population cannot be estimated, a population index can be very valuable. This is a statistic assumed to be related to the population size and reflective of any change in population. Another option is to use a surrogate, which is a variable that is assumed to be representative of another variable, and it should have a strong relationship or causal link to the population. A good example of all three of these is shown in this graphic for Fisher. The dark blue line is the fall population estimate. The red line is the winter track index. That shows the percent of survey segments with Fisher tracks present. And you can see that it matches pretty nicely with the population estimate. And then at the bottom, we have the pink line, which shows the harvest numbers. So you could use harvest as a surrogate for the population if you can assume that when the population increases or decreases, the harvest increases or decreases. And you can see that the trend kind of follows the population trend, though not as apparent as the change in population or winter track index. So there's more that goes into a surrogate measure than that needs to be accounted for. So choosing the right metric to be able to detect the amount of change that is necessary to manage a species is a really important consideration. Some of the methods for sampling populations include mark and recapture or recite. In this case, the population size can be calculated by multiplying the number of individuals in the first sample, like this sample of 10 turtles, multiplying that by the number of individuals in the second sample, and then dividing by the number of individuals in the second sample that were recaptured. This is a pretty common method used to estimate population size. Another method is distance sampling, which uses the concept that the probability of detecting an animal decreases as its distance from the observer increases. It uses the detection probability to account for individuals that were missed, improving the precision of the estimate and reducing bias. Detection probability, so that's shown in this graph on the bottom. Another method is the complete plot count, which is when a set plot size is fully counted and then the estimate is extrapolated to all the potential plots across the landscape. Without the incorporation of other covariates, this method assumes that population density is consistent across the landscape, which is not often the case. And then a lot of what goes into population estimation is population modeling. This can take into account many different covariates and estimates to model the population under different conditions. Models inherently have uncertainty. So like in this example here in the right bottom corner, you've got a number of different models for the bear population compared to each other, and also compared to a mark recapture estimate, which is in orange. So, like we've seen, there can be a lot of uncertainty and bias in population estimation. Some of the other disadvantages are that the methods typically require capture, marking, observation, or sighting of wildlife. This can be difficult for species that are small, secretive, nocturnal, quiet, or at low densities. Estimating detection probability is typically required to understand whether the species is absent or just undetected. And some of these methods are typically costly and not always practical, especially when you have many covariates that can impact the population. They're also difficult to collect over large areas due to the logistics and effort. And measuring the population alone doesn't answer why. I mentioned covariates a couple times. These are variables that influence the population or the detection probability and can be pretty much anything from weather, such as rainfall, winter severity, snow depth, habitat components like cover and food abundance, parasite and disease prevalence in a given year, and harvest level. Measuring covariates can help to determine why changes in population metrics are observed. Population indices are a good alternative when the population as a whole cannot be estimated. They're typically lower cost and easier to collect. Some examples are harvest data, sign surveys, and camera traps. A common method is to calculate the observations per unit of effort or area. But one must choose a metric that is sensitive to population change, and there's always potential for bias or misinterpretation. A good example is the Shartell grouse survey. The index that we use is the mean grouse per lek surveyed. And if you look at the statewide estimate of this index, it looks like things are relatively stable over the past 10 years. However, if you look at a different index, which is the number of leks counted, and you break it out by region, you can see that there's quite different trajectories for the Northwest and the East Central populations. So one metric on its own is likely not giving you the full story. And we must be cautious in interpreting single metrics without more information. Another good example of that is the wolf population. There are quite a few different metrics that go into modeling the wolf population estimate. If you look only at the number of wolf packs in the bottom left, you might think that the population is increasing. However, the population is relatively stable in the estimate, and that difference can be explained by a reduction in the average territory size and the midwinter pack size of wolves. So how do you know when you have all the right metrics? Designing population surveys may seem simple. Determine the question or goal, define the population of interest biologically, spatially, and temporally, choose metrics and covariates to monitor, design monitoring that can detect population change, taking into account statistical power necessary to detect change in the acceptable size of confidence intervals, and then adapt within the given constraints of budget, staff, access, and feasibility. So as an example, I'm going to go over our annual moose survey. The goals of this survey are to determine the population's long-term trend, whether it's decreasing, stable, or increasing, as well as the composition and spatial distribution of the population, and then assess if harvest is sustainable and set harvest quotas for the hunting season when it's open. And then those findings will be combined with research to improve our understanding of moose ecology and ultimately contribute to future population and habitat management strategies. So the northeastern moose range is nearly 4 million acres. How would you go about monitoring or estimating the population? What the Minnesota DNR does is calculates a population estimate to monitor change in the status and also estimates age structure using percent calves, calf to cow ratio, and bull to cow ratio to identify any potential issues with reproduction. The Northeast Moose Range is broken up down into 436 survey plots. Approximately 10% are sampled each year, plus 9 permanent habitat plots that evaluate the effect of forest disturbance. Flocks are surveyed by helicopter, counting the number of moose observed by age and sex. Getting a little more technical, we must account for visibility bias using a sitability model. Radio collared moose were used to estimate the probability of observing a moose when it was present and how the amount of visual obstruction from vegetation affects that detection. After correcting for detection, the estimate is then adjusted based on the sampling methods, in this case, extrapolating from the surveyed blocks to the entire Northeast range using models. Confidence intervals around the population estimate are calculated, and trend lines are fitted to try to explain the population trajectory. Here's the latest results from the moose survey over the past 15 or so years. The results are best suited to establishing long-term trends. Short-term trends must be viewed cautiously. As you can see, the confidence intervals from year to year often overlap, suggesting there's no statistically significant change from year to year. However, the long-term linear trend shows a decline. But while the linear trend is declining, if you fit a more complex piecewise polynomial curve, it indicates a decline and then a more recent stable trend. So the interpretation of the data can be quite different based on the analysis methods and how you look at it. Another example of how we monitor wildlife populations is ruffed grouse. In this case, we do not try to estimate the entire population size. Instead, the spring drumming survey calculates a population index. There are routes with 10 stops each surveyed across the ruffed grouse range, and the number of drumming grouse heard at each stop is recorded. The DNR reports the number of drums per stop annually and monitors the change in this index over time. Ruffed grouse populations are known to follow a 10-year cyclical pattern of highs and lows, so it's expected that we should see a cycle of highs and lows in the population index. And that's exactly what we see when we look at the data from the past 50 years. Generally, the highs and lows are similar around the average over time, suggesting that the statewide population is stable. So if the population index says the population is stable, why do hunters report and harvest fewer birds? The graph in the left corner shows the decline in harvest numbers over the past 20 years in gray. Could it be due to the fact that hunter numbers are also declining, or are there fewer younger birds to harvest in the fall? Findings like this spark the need for further research, and in this case, the DNR has started to conduct brood surveys, quantifying the number of broods observed through the summer per unit of effort, in this case, field staff working and driving in the woods. Multiple years of data are needed before a trend can be observed. So that work is ongoing. Another step in trying to explore ruffed grouse population change is the effect of West Nile virus. The DNR sampled hunter harvested grouse and found that West Nile virus is present in Minnesota, but grouse can survive exposure. How West Nile virus might affect broods prior to the fall is still a mystery. Clearly, there are many challenges in monitoring wildlife populations. Determining cause and effect, teasing apart complex relationships, measuring the right metrics, interpreting the data appropriately, and the usual constraints of funding, time, access, equipment, and infrastructure make uncovering the full story a real challenge. So how can we tease apart all of the factors that may affect a population? And how do we know when habitat is a factor? Research and monitoring are both needed. Take moose, for example. They're impacted by a number of things: climate change, wolf predation, parasites and diseases, habitat availability, habitat quality, interactions with deer, and reduced reproduction. How all of these different factors are connected and where we can use management to conserve moose is a big question. Would habitat management be enough? Measuring wildlife habitat is an important part of the story. It can inform wildlife population monitoring and research. Habitat metrics can be used as indices to the population. Typically, however, you cannot measure all aspects of habitat at once. In order to use habitat as a population index, you need to establish relationships and preferences, which require research investment upfront or well-established existing knowledge. You also need to be able to quantify habitat components, which can be quite complex. One example is marten use of den and rest sites in some standard forest metrics. Marten use sites with a higher volume of CWD and higher surrounding sapling density, though that is only a very small piece of the habitat preferences of marten. And how could we then take that information and use it to identify habitat? We really need to know more of the story. Some of the benefits of measuring wildlife habitat is it can often be easier than measuring wildlife populations directly. A number of the components can be remotely sensed or use existing forest inventory data sets. Habitat data is often easier to collect in the field, and it doesn't require wildlife detection, marking, or capture once habitat relationships are already researched and defined. But habitat is not always occupied, or the resource is not necessarily available. This is one of the biggest challenges to trying to use habitat in place of monitoring wildlife directly. Habitat relationships can change over time and be influenced by many other variables. Using habitat information in combination with wildlife population data can help us answer complex questions and fill in a big part of the story. A good example of using a habitat metric to help inform wildlife management is the Bear Food Survey. This survey measures just one component of bear habitat, which is food. And the analysis of this data has found that fall food production, in this case, dogwood, oak, and hazel, along with hunter numbers, can be used to predict the bear harvest, which is an important component of predicting the bear population. Another example is the moose habitat component of the survey. This project is led by Fond du Lac biologist Mike Schrag, and it measures moose density in permanent plots where disturbance has occurred and tracks the use of these areas by moose over time since disturbance. In some cases, we also have pre-disturbance data available. And this project incorporates timber management plots, prescribed burn plots, and wildfire plots to learn more about how moose use these areas following disturbance. Forest habitat is a phrase often used incorrectly to refer to cover type and age class of the forest, and I am guilty of that myself. Habitat is not habitat type, vegetation type, or forest cover type, but we can use forest cover type and other forest metrics as surrogates for habitat, and even further for surrogates for wildlife populations. Forest cover type is a low-cost metric. There's lots of data available already, and it can be continuous across the landscape. However, there are many challenges and assumptions that must be acknowledged. Species must be tightly linked to the vegetation. This is often not the case because there are usually complex relationships to variables beyond cover type, both forest metrics and other factors. Here's a good example here. Looking at aspen by age class in Itasca County from FIA data. One might interpret this to suggest that there's more young aspen in the 11 to 30 age classes, and thus there should be more ruffed grouse habitat. But then why do we not have more ruffed grouse? It's just not that simple. Monitoring populations is only the beginning of trying to understand what drives wildlife populations and ultimately how to manage them to reach a desired outcome. A good example of the role research must play in understanding species and habitat interactions is our winter deer habitat study. The goals of this research are to improve our understanding of white-tailed deer winter habitat relationships at the stand level relative to winter severity and deer density, and then to formulate habitat management prescriptions that better support population goals and are more easily and effectively integrated with long-term forest management strategies. The winter deer survival rates rely on minimizing energy expenditures, especially in winters with deep snow and extreme temperatures. The arrangement of different types of cover is important for this. Closer proximity of cover and browse, which is food, allows for less energy expenditures moving between the habitat needs. However, deer density can negate that if there are too many deer using the resources. So we have a need to quantify the relationships between deer survival and deer health and cover and browse arrangement, while also factoring in winter severity and deer density so that we can plan forest harvest in a way that will best benefit deer. This study is being done at 2 sites that are known winter deer yard locations: the Ingwadonna Lake study site near Reamer and the Elephant Lake study site near Orr. Deer at the study sites are fitted with GPS radio collars that transmit their location every 2 hours. So we're getting very fine detailed habitat information that can be used to help tell the story of deer use of cover and browse. At these study sites, habitat classification and inventory is done via photo interpretation to accurately describe the cover and browse potential. Biologists identify conifer density, browse availability, and other habitat features. And all this information goes into a complex analysis of the location and movement data from the GPS collared deer, as well as monitoring their nutritional status, survival, and causes of mortality. So with all of this in mind, how should we think about wildlife population and habitat goals? Population abundance, harvest numbers, amount of habitat, and other discrete measures aren't the goal, because operating in that way in a changing world is just not practical. Those metrics are targets and indicators that must be adaptable. The goal, like the goal from the moose management plan, is to the greatest extent possible manage for ecological sustainability and hunting and viewing opportunities. Another example of wildlife population goals from the ruffed grouse management plan, and that's to ensure the viability of the ruffed grouse population and their forest habitat, to manage for ruffed grouse as an integral part of Minnesota's forested landscapes, and to encourage and promote recreational opportunities for the enjoyment, hunting, and observing of ruffed grouse. And actually, this was just an excuse to put a picture of my dog in the presentation. So what does that all mean? Things change. Targets for population size and harvest must be dynamic and ever-changing. Population metrics are necessary as indicators to help guide our work, and monitoring populations, habitats, and other variables is necessary to unravel the complex interactions and effects that influence how we manage wildlife and habitat. Altering management and expectations to best reach our desired outcomes should be the standard practice. Another challenge we face is balancing multiple species goals. Many species are intricately linked through complex ecological relationships, both positive and negative. Management for one species must consider impacts and limitations imposed by others. We must acknowledge that trade-offs are necessary. The deer plan demonstrates that with their guidance on deer in the moose range. The direction is to manage deer in the moose range at levels consistent with the moose plan, recognizing that moose are a species of special concern and they will be the priority for big game population in that and habitat management in those areas. This need for a trade-off is also true for wildlife populations and habitat and other forest resources such as timber. So what are the major takeaways from today? First is understanding wildlife populations and their habitat needs is not easy or cheap. We need to invest in monitoring and research as well as adaptive management in order to further our understanding of how we might be able to influence wildlife populations with habitat management. Second is that complex relationships hinder the ability to just measure forest metrics instead of habitat and populations. The more of the story that we can understand, the better we can design management for wildlife and their habitats. And finally, we must work together to balance multiple goals in the forest. Trade-offs are necessary, and we should be discussing and acknowledging this and working together. With that, I'd like to acknowledge the Forest Wildlife Populations and Research Group and our scientists who take on this challenging work every day. Glenn Delgidis, John Erb, Charlotte Roy, Andy Try, Carolyn Humpel, and recently retired Dave Garcelis. You can find out more about our surveys and research on the Minnesota DNR website by searching for wildlife research publications. Thank you.